Thank you very much for joining to this webinar that we are organizing. And today's topic is going to be about new technologies to study cell migration. So for your information, this webinar is being recorded. So in the end, if you want to have access to it, we can share it with you. And so just for you to understand how it's going to be, so first we are going to have a presentation and at the end a session of questions and answers. But meanwhile, you can type your questions on the Q&A option. And later on, if you want to ask a question directly, I can give you access to the microphone and you can ask your question and then we proceed from there. Okay, so my name is Katja. I'm the application scientist at 4DCell. And I will be one of the speakers today, together with my colleagues, Patricia Davidson, our R&D project manager, and Rita Ribeiro, our CTO. So 4DCell is a biotech company located in Paris, France, and we develop in vitro technologies or assays with the aim of controlling the cell microenvironment. And our idea is to help scientists to obtain more relevant and accurate readouts by using this type of assays. So here you can see on your right picture of our facilities and the clean room where we produce or fabricate our products that we send to our clients. And also a very happy Patricia here working with some microchannels. So you see how it is in the clean room on a regular day at 4DCell. So I'm going to start by giving you a brief background on cell migration. As we all know, it's a very important process involved in physiological events such as immune responses and tissue homeostasis like wound healing, embryonic development. But in addition as well, cell migration has an important or is very relevant in terms of pathological conditions, for example, in chronic inflammatory diseases and in cancer, right? So then different cell types, they migrate differently depending on the type of cell they are and their function. Important to remember is that, of course, they can move individually or collectively. And the two main types of migration described in the literature are termed mesenchymal migration, as I have here, and amoeboid migration. In the case of mesenchymal migration, this is more typical of adherent type of cells, such as, for example, fibroblasts and muscle cells, whereas amoeboid migration is more commonly seen displayed by non-adherent cells, such as immune cells. And of course, to do experiments in a controlled microenvironment, it's very important in order to understand how cell migration works. We all know that. And for a long time, the molecular mechanisms underlying cell migration have been studied mainly using 1D and 2D type of substrates. So actually, we are going to show you some examples of this type of substrates that you can use in these studies, such as, for example, micropatterns, dynamic or another option. But although the micropatterns can be used to mimic certain conditions in vivo, for example, in the case of cells encountering lining body cavities, they do not entirely mimic, right, the in vivo microenvironment, the complexity. Therefore, we wanted to show you as well some another set of tools such as the micropillars, microchannels, and the cell confiner, because they have or they provide the more to 2.5 to 3D type of matrix, which resembles better the complexity of the in vivo microenvironment. And with this, I will pass the word to Rita, my colleague, and she will give you some features and descriptions about the different options of products we have available for cell migration.
Thank you, Katja, for the nice introduction. Good afternoon or good morning, everyone. So during the next few minutes, I'm going to describe the technologies we have available at 4DCell and that can be used to study cell migration. We are going to start with the micropatterns. If you can, thank you. So micropatterns are basically substrates that are chemically modified, where you have areas where cells can adhere specifically. So you have a non-adhesive coating around these areas. These areas where cells can adhere, they can be bare glass or they can be coated with some extracellular matrix, some ECM matrices. For cell migration, typically these substrates, they have the form, or these patterns, they have the form of lines so that you can observe cells migrating along these lines. So these are our standard micropatterns, but we also have another type of micropatterns where the non-adhesive coating can be switched into an adhesive coating at some point, and you can study the evasion of cells towards this area that was non-adhesive at the beginning. At the bottom, you can see just a cross-section of a patterned cell, and on the next you will be able to see some C2C12 cells patterned on line micropatterns. These devices, the micropatterns are available in different formats, both the glass cover slips and the glass bottom Petri dishes. And then you have multi-well plates, either with the 96 wells or 384 wells. Otherwise, if you want to do it by yourself, we have fabrication kits that we can provide you, and you can pattern your own glass cover slips. So this was the first technology, more towards 1D cell migration. Now we are going to show you some details about our microchannels. Microchannels are simply microfluidic devices made of an elastomer. They are quite simple, and the principle of use is quite simple. So they are just some small chips composed of bundles of microchannels connected by access ports. You see the cells on these access ports, and they will migrate between them in these microchannels. And microchannels are basically simple tunnels, as you can see in the next picture, in the cross-section on your left. You can see that these channels are simple tunnels where cells go through. And in the cross-section of the right, you can see that the channels, they connect both access ports on their left and on their right. The microchannels can be found in two shapes, let's say, straight microchannels where cells can simply migrate inside, or microchannels containing constrictions, so cells will encounter some sort of obstacle like they encounter inside our body when they are migrating. On your left, you will see now a picture of a HeLa cell migrating towards one of these special microchannels containing a constriction, and you can see the scary face it is doing if you can look at it. So the microchannels that we are doing, we are using some very high resolution modes to do them. You can see here some scanning electron microscope images. And here is an example of the smallest constriction we have available that has a width of 1.5 microns and can have a height of 5 microns, so with a quite big aspect ratio. The microchannels that we have that we provide at 4DCell are available in 35 millimeter glass bottom Petri dishes. But as the micropatterns, you can also have a fabrication kit that you can use at your own lab to produce them by yourselves. And you can, if you want, customize the design of the microchannels. The third technology that we are presenting here today is based on structured surfaces. In this case, it's a substrate containing topographical features, micropillars, and these micropillars will have different diameters. Depending on the diameter that they have, they will give a feeling to the cell that it is on the top of a substrate with a specific stiffness. So larger pillars are stiffer than smaller pillars. So the cell will migrate towards where it's more comfortable, let's say. So in the following image, you just see a scanning electron microscope image of a cell on the top of two different types of pillars. So this image is a bit impressive because it's a scanning electron microscope image. Of course, is not what you acquire when you do optical microscopy. but it illustrates a bit what I just mentioned. The micropillars are provided in 10 millimeter glass cover slips. And following this, we have another type of structured surfaces. These are a bit like microchannels, but open microchannels with the migration application in mind. So basically what we have, it's glass cover slips coated either with a soft or hard material. So it can be an hydrogel or an elastomer. And we can go from very soft substrates like 1 kilopascal to 1 megapascal or even higher. And when you seed your cells, the good point about this is when you seed your cells, they will be directly on the top of the structures. So rather than in the microchannels where you need to seed your cells in the access ports and wait for them to enter the microchannels and start migrating, in these devices, you seed your cells on the top of the microstructures. The cells will prefer some of them to be in the grooves area, in these line-shaped grooves, and they will start migrating. So the process is faster. These devices, you can see here at the bottom cross-section of what I just mentioned, and these devices are typically provided in 24 millimeter glass cover slips, so something quite standard. The following device, and the last one that we have to show you here today, the last technology, it's the cell confiner, and it's very special to us. So the cell confiner is basically a device that we have that enables to confine cells in between two parallel plates. The normal heights that people use to confine cells are between 1 and 20 microns, but you can also opt for higher pillars. How does this work? It's a very simple process indeed. So basically you have a cover slip that has micropillars on it. So it's a microstructured cover slip that you attach to a transparent piston. It can be made of a PEMS. And after you simply put this assembly on the top of your cells, and it will confine your cells to the height of the pillars that you have predefined. So this can be used, for example, to study migration of immune cells that are usually migrating slower than what they migrate if they are inside the human body. So if you confine them with if you use this device to confine them, you will see that the migration dynamics will change quite a lot. On your left, you can see some HeLa cells confined and not confined. So you can see the blabbing on the right that occurs when cells are confined at 3 microns, for example. The cell confiner is available in 2 formats. The static confiner, that is controlled by the user, and the dynamic confiner that is controlled by a vacuum pump. So how does this work? In the 1-well static confiner and in the 6-well static confiner, and the first is compatible with Petri dishes, and the second with the multi-well plates, 6-well plates in this case, you basically have like a modified lid where you assemble your substrate, your cell culture system, and then by closing it, you confine your cells. On the other side, the dynamic confiner is a bit more complex, and you have a suction cup that is connected to a vacuum pump that basically makes the piston go down and confine the cells that are between the confinement slide. So the dynamic confiner can be very useful if you want to image the moment you are confining your cells. It's because you can control this process much better than if you do it with static confiners. That can be quite useful if you want to study confinement of cells a long time, and if you want to see what happens after confinement. So now I'm going to pass the word to my colleague Patricia, who is going to speak about some of the specific applications using these devices for cell migration.
Thanks, Rita. So as Katja mentioned, I'm a R&D project manager at 4D-Cell, and so I'm going to walk you through some of the things you can do with the devices that Rita just mentioned. So the very first one is perhaps the simplest way you can study migration just in 1D. So at 4D-Cell, we offer micro-patterned lines of different sizes on the same cover slip. And so here you can see I seeded fibroblasts on lines that are, in this case, 10 microns wide. And you can see they're very nicely elongated. And it turns out that in cells that are on 1D lines, they have a phenotype that's very similar to cells migrating in 3D through an extracellular matrix. So there are publications that describe the fact that cell migrating on 1D lines are a better technique to imitate migration in vivo than 2D migration. So with our micropatterns, I was able to acquire this beautiful movie using our holographic microscope. And you can see that the cells are migrating along the lines, and the micropatterns are holding up quite well. Sometimes the cells are even able to skip from one line to another. And so with this, you can measure the migration persistence and the velocity, for example, or any other application you can think of. Another type of micropattern that Rita already mentioned are the dynamic micropatterns. So in this case, in this example, I seeded cells on a 500 micron circle. And so the cells become trapped in this micropattern, but you can add a protein via click chemistry to the surface. Basically, the protein we're adding is a small part of fibronectin. And so when you add this, this triggering reagent, the cells are able to migrate out of the micropattern as if the rest of the surface was coated with fibronectin. And you can see with time that this circle becomes larger as the cells are migrating out. So again, you can measure cell velocity. You can quantify it using plugins in ImageJ, for example. And again, you can acquire nice movies. And as we have several micropatterns on a cover slip, you can get statistics on your cells. So here we can see several circles growing bigger after we added the triggering reagent. So now I'm going to talk about some 2.5D surfaces. As Rita mentioned, they're not 2D, but they still have topography in 3D, so we call them 2.5D. The first one I'm going to talk about are these surfaces with grooves. So here we have grooves that are 10 micron wide and 15 micron tall. In this case, the mouse embryonic fibroblasts prefer to stay on the flat surfaces on the top above the grooves, whereas we know that HeLa cells, for example, will burrow into the grooves, as shown in the picture on the bottom. So you can really have different phenomena given the different cell types that you try. Importantly, these substrates are made of an elastomer, PDMS, which is quite stiff, like a rubber, but we can also make these out of hydrogels, which will have different stiffnesses. And so you can imagine that you could make substrates that would imitate, for example, the brain, which is quite soft, or substrates that would imitate the bone. So we can make any shape you would like in a 3D surface. So basically anything that you want to imitate in vivo, we can give it a try. It's just your imagination is the limitation. So the next surface I'd like to talk about are the micropillars. So as Rita mentioned also, the stiffness of the micropillars depends very strongly on the diameter of the micropillars. So by varying the diameter of the micropillars, you can get very different surfaces for the cells to interact with. And in this case, we can study durotaxis, which is the ability of cells to migrate towards or away from stiffer surfaces. In this study, they showed that rat embryonic fibroblasts will actually migrate from a soft substrate towards a stiffer substrate. And you can see in the bottom images that they're migrating towards the larger micropillars. So at 4D-Cell, we offer similar substrates. So the smaller pillars are 2 micron in diameter, and the larger ones have 4 micron in diameter with a 6 micron height. So they're quite high aspect ratio. So we can also look at cell migration in 3D under confinement. So the first product I'll talk about are the microchannels. Here you can see two types of cells migrating through microchannels. I have HeLa cells and mouse embryonic fibroblasts. And you can also take very nice movies of cells migrating through. Here are dendritic cells that are quite fast at migrating through channels. And so you can compare their speed and their persistence in channels of different sizes. So here, it looks similar to the 1D migration, but in fact, the cells are confined from the sides and on the top and bottom. If you want to restrict your cells even more, we also have microchannels that have these constrictions. So in this case, a 3-micron constriction and an 8-micron channel. And our client uses these constrictions to show that lymphocytes, which are also very fast migrating cells, lose their ability to migrate through 3-micron constrictions when you knock out hormone like 1. So this phenomenon is very likely related to the nucleus. And so we have observed nuclear deformation in our constrictions. And if you'd like to know more, stay tuned for a future webinar on the nucleus. So now I'm going to pass the torch over to Katja.
Thank you, Patricia, for passing the word back to me. Yes, I'm going to give some examples of scientific applications connected with the cell confiner. For example, migration phenotype transition using eukaryotic cells. So the confiner, this particular application is very interesting, for example, for researchers working with cancer, and particularly tissue invasion mediated by cancer cells. And one interesting observation of this study is that even if the cells are non-migrating type, the fact that they are confined, and particularly when in contact or adherent to a specific type of substrate, the authors saw a shift on the migrating phenotype the cells were displaying upon these conditions. And as you can see here, the more compressed the cells are, the more they shift to a migrating type of phenotype. One also important observation from this study is that with the cell confiner, it's possible then to evaluate the interaction between the cell's microenvironment and their migration behavior. So in line with this application, I'm going to show you another one also regarding migration phenotype transition. And this is a paper by our client Thibault. This was recently published. This application, in this case, shows the migration phenotype transition in flagellate organisms instead of eukaryotic cells or mammalian cells. And what Thibault observed, and you can see that as well in this image, is that the flagellates, when under confinement or in confined environments, they acquire an amoeboid-like phenotype, which is very prominent, as you can see here. And he also then described in this paper that this amoeboid type of phenotype the flagellates acquire is conserved across animals and flagellates themselves, which it's very interesting from the evolutionary point of view, of course. So also recently, last year, two of our clients, Verena and Stefan, they also published a very nice paper in Science, where they show that by using the cell confiner, cells can be deformed in a way that, excuse me, that the deformation of the cells defines their contractility, polarization, and also a switch to a more amoeboid cell type of migration, as you can appreciate in these graphs here. And they described the mechanism to be mediated through the cell nucleus, which is acting as a mechanosensor in this case. The last application I would like to share with you that can be achieved using the cell confiner, and this is also very interesting for those who work with the immune system and for example, with chronic inflammatory diseases, whereby there is a recurrent recruitment of immune cells to the site of injury, is that once you confine immune cells, and this goes along with what Rita said in the beginning, once the immune cells are confined, the authors observed that there is a faster migration, so the cells move faster, and they are also more likely to interact with the other cells in the vicinity. As you can see in this video, these are dendritic cells in a non-confined environment, normal Petri dish. And in the second video, you see the dendritic cells upon being confined. And you can see that they move much faster and they basically interact with the other ones around. This is translated in this graph where you see the diffusion coefficient of the cells, which is much like 5 times higher once they are under confinement. So with this, I wanted to just to move on with a reminder that if you would like to attend our future webinars, we have now 2 scheduled: cell differentiation by the end of May and wound healing in the end of June or mid-June, at the same time as now, 4:30. And you are welcome to join and register. And so now with this, I closed our presentation, and I will introduce the question and answers session.